Okay, so welcome to our lecture on bacterial anatomy and requirements for infection. So you may ask yourself, well, why should we know about bacterial anatomy? You know, it's very easy for everyone to think of why human anatomy is so important to know and understand, but bacterial anatomy is just as important. You're going to see that throughout today's lecture and honestly throughout the course as a whole, because understanding the concepts in this lecture will help us to target various infections and better understand how to treat patients who have any kind of problem in terms of bacteria invading. Now, before we go into the specifics of the anatomy and infection aspects, the first thing to remind you is the difference between prokaryotes and eukaryotes. So again, always think no. So prokaryotes are any cells that do not have a nucleus and do not have any internal membranes, whereas eukaryotes are like our cells, which have a nucleus and have internal membranes. Now, one of the things I'm going to keep pointing out in today's lecture is to make note that fungi are eukaryotes. Okay, so put a little star next to fungi being eukaryotes, because so many times I hear people and students basically refer to fungi such as yeast or mold as if they're bacteria. And that is very, very different from fungi, okay? Because fungi have a nucleus, they're eukaryotes, so in fact they're more like our cells than they are like bacteria. So please keep in mind that fungi are eukaryotic. So on this slide, we review the domains and kingdoms, and a lot of this, again, this early portion of the slides is more like a review for things you've probably encountered elsewhere. So the three domains, which you kind of saw a little bit of on the last slide, are bacteria, archaea, and eukarya. And when we talk about bacteria and archaea, they are both prokaryotic, whereas eukarya, of course, are eukaryotic. And we just reminded you what exactly prokaryotic means. So again, the idea that bacteria and archaea do not have nuclei and do not have internal membranes. Bacteria is one of the biggest categories that we're going to talk about throughout this semester in this course, whereas archaea we don't encounter as much, but they are still very important to know, because archaea are these cells that are capable of surviving in extreme environments. So basically picture these organisms that can live in a volcano or deep within the earth. Okay, these guys are very important to us because if they're capable of surviving these extreme environments, then that means that they have enzymes and proteins that are capable of surviving these extreme conditions. And we've actually isolated some of them to use in research in the medical field. One of the most common ones that you encounter is Taq polymerase. So if you've ever heard of PCR, which we're going to talk about in a later chapter, we're able to perform these experiments with Taq polymerase because we can have this enzyme that withstands very high temperatures because it was isolated from archaea. Now the last category here, the last domain, is eukarya. And again, keep in mind eukaryotes, including our cells, have nuclei and they have internal membranes, including various organelles. Now, once again, I want you in this figure over here to put a star and circle fungi as eukaryotic. You can even put a little star underneath eukaryotes here and write fungi such as yeast and mold are eukaryotic, meaning they have a nucleus. So again, fungi falls into eukaryotes and it tells you that they have a nucleus. These three slides that we're going to see now, these are basically just a review of information that we talked about on the last slide. You don't have to memorize anything that you find on the side here. It's just to show you what exactly we mean by bacteria. Then we have a visual of archaea. And again, the only things that I want you to remember for archaea is that they are prokaryotic, so there is no nucleus. They can survive in extreme environments, and we can obtain valuable enzymes from them. And then the last one here, we have the eukaryotes, which includes us. And again, notice that fungi is included as well. And anything found on this slide should have a nucleus and internal membranes as organelles. Now, as we're considering the anatomy of bacteria and microbes, one of the things you'll have to understand is the various shapes that bacteria can be found in. There are three main shapes that we talk about for bacteria, even though you kind of may sometimes see them listed out in a few more extra shapes. The three main shapes that we always refer to are coccus, which is round bacteria, bacillus, which tells you it is rod-shaped, and spiralia, which is spiral-shaped. Okay, so make sure you know coccus, bacillus, and spiralia. And then the shapes, especially coccus, can be arranged in different ways. So when you gram stain some cells, or you look at some cells throughout this course under the microscope, their arrangement is basically how they line up or how they congregate together. So some of the arrangements we have over here in the corner. You can have diplococcal arrangements, which means a pair of cells. You can have a tetrad arrangement, which is a set of 4. And then you have streptococcus versus staphylococcus. And I want you to put stars next to both streptococcus and staphylococcus, because these will keep coming back throughout the semester, especially since two of the biggest genuses that cause infections in humans are strep and staph. And we're going to kind of study them a lot more in the lab setting. But I want you to remember, streptococcus arrangement, as you can see here, is a linear arrangement. We always remember that as a strip of strep, okay, or strep strip, meaning a line. Whereas staphylococcus, which comes from Greek for grapes, looks like a cluster of grapes. Okay, so you can think of a staff of nurses clustered together. Okay, so strep is always a linear arrangement, whereas staph is a clustered grape-like arrangement. Now again, we're getting back to the idea of bacterial anatomy. And as I mentioned in the first slide, you have to ask yourself, why study this anatomy of the bacterial cell? So for that, like we said, it helps us understand what to target to kill the bacteria. And it also helps us understand how exactly each type of bacteria is making us sick. So it's very valuable to understand the anatomy of a bacteria. And one of the structures that's going to keep coming up throughout this course is the bacterial cell wall. Okay, so I want you to circle cell wall. Put little stars next to that, because that is a structure that bacteria have, but we do not. And that's going to keep coming back as very important throughout this course. So, as I just mentioned, the most important structure to remember is the bacterial cell wall. Again, this is a structure that bacteria have, but we do not. which is going to come up a lot in this course because that makes it an excellent target for when you're trying to treat a patient. Okay, so when we talk about the bacterial cell wall, we categorize bacterial cells into two different categories based on their cell wall composition. So there are gram-positive cells and there are gram-negative cells. Gram-positive cells, you'll notice, they have a thick layer of peptidoglycan. And what peptidoglycan is, if you remember from one of our previous lectures, that is a carbohydrate structure. Okay, it's carbohydrate with some peptides thrown in as a network. You'll see a picture of it in a slide or two. There's also their plasma membrane, which we also have. Okay, so notice all cells have a plasma membrane. And then for gram-positives, there's teichoic acid sticking out, and we'll talk about that soon with regard to giving certain cells a bit of extra toxicity and also sometimes helping with attachment. Now, what you'll notice when you then look at gram-negative cells is these two cell walls look very different from each other. Gram-negative cells now have a very thin peptidoglycan layer. They then have an extra gap and an outer membrane. And their outer membrane has a lot of lipids and very selective porins. So what I want you to notice is when you look at these two cells, which one do you think is tougher to treat if someone has an infection? Well, which one would it be harder to get antibiotics into or drugs into? Gram-negatives, right? So put a little star over here and write that gram-negatives are not as sensitive to antibiotics and drugs because they have these extra layers and they have very selective porins, okay? Very selective porins. So gram-negatives are more resistant, tougher to treat, whereas gram-positives are more sensitive to antibiotics. The other difference that I want you to know is that gram-positives tend to release what we call exotoxins, whereas gram-negatives tend to release endotoxins. Okay, so we're going to talk about these in more detail at the end of this lecture. But for now, know that positives are associated with exotoxins, whereas negatives are associated with endotoxins. Okay? And a little trick to help you remember the difference between the two is I always think to myself, gram-positives are the ones with plenty of peptidoglycan. Okay, that P sound. So this slide is simply to review what I just went over on the previous slide. So first of all, the idea that bacterial cell walls have peptidoglycan, which is a network of sugars and of peptides. You'll see more pictures on the next slide, but this is just one small figure to show you the two sugars that are the main component of peptidoglycan. So they're NAG and NAM. There is a whole lot of this peptidoglycan in gram-positive cell walls. And remember, gram-positives, we said, are the more sensitive of the two types of cells. And they also tend to be associated with exotoxins. Whereas gram-negatives, these guys are the ones with less peptidoglycan, but a whole lot of lipids. Okay, put little stars next to that because it's going to be important in later slides as well as in lab, that gram-negatives have a whole lot of lipids. And then put a star next to the fact that gram-negatives are more resistant, so they're going to be tougher to treat, and this is due to those selective pores we mentioned, as well as the extra layers that they have. And the last little point there is a reminder that gram-negatives tend to be associated with endotoxin. This slide here is simply a figure to help you visualize what we mean by peptidoglycan and by cell wall structures. So this shows that the peptidoglycan is a network of those sugars as well as some peptides thrown in. And then you can see the difference in the layers of a gram-positive versus a gram-negative cell. Okay, but again, you never have to memorize any of these kind of figures. It's just to help you visualize. Now, when we talk about the bacterial cell wall and the different components of the gram-positives and gram-negatives, I just want to point out a few clinical significances in terms of those components. So with gram-positives, the fact that they have teichoic acid helps them to stick within your nasal cavities. So a common one to do this is Staph aureus, if you've heard of staph infections. And when I write that nosocomial staph aureus infections do this, nosocomial, remember, simply means infections picked up in hospitals. Okay, so the teichoic acid helps the bacteria stick to the inner linings of your nose, and the teichoic acid also gets released when any of these bacteria dies and triggers an inflammatory response. So basically, the teichoic acid breaks away, and within your body, your immune system recognizes it as an invader and triggers inflammation and the immune response, which can be problematic. You then have the gram-negatives, and the clinical examples for gram-negatives is that, first of all, that outer membrane that they have, that makes them, like I said, more resistant, which means that it's going to help protect them from antibiotics and disinfectants that you may use to try and clean areas or treat patients. And they also have lipids that include lipid A, and when the gram-negative cell dies and releases lipid A, this causes a major immune response, okay, and can basically show a lot of symptoms in the patient, especially inflammation and cell deterioration. Some examples of cells that do this are Salmonella, Shigella, and E. coli, all of which we're going to talk about in more detail in later lectures. Now, after spending so much time pointing out the significance of bacterial cell walls and all of these bacteria that have it, I now want to make note that there is one type of bacteria that does not have a cell wall. And I want you to circle, star, highlight the name Mycoplasma. Okay, Mycoplasma. This is the smallest of bacteria, but what is most significant about it is that these guys do not have a cell wall. An example of mycoplasma is walking pneumonia. Now, it's very important to know that they have no cell wall, because now you have to ask yourself, well, why is that significant? What did we say about bacterial anatomy? Knowing bacterial anatomy lets us know how to treat the bacterial infections. And when we do our chapter on antibiotics, you're going to see that a lot of drugs that we give patients, a lot of antibiotics, they target cell walls, including the most famous one, penicillin. If your patient is suffering from walking pneumonia or mycoplasma infection, what's not going to work on them? Anything that targets cell walls, because these bacteria do not have a cell wall. Okay, so for the question, why is that significant that they have no cell wall? You can write, because they cannot be treated by antibiotics such as penicillin that target the cell wall. You must use different antibiotics. Okay, so in addition to having the cell wall, some bacteria may have some other structures outside of the cell wall. So ones to remember are the glycocalyx, fimbriae, pili, flagella, and axial filaments. And all of these you'll hear at various times throughout the course. Now for glycocalyx, there's a few facts that I want you to remember. First of all, the glycocalyx can either be firmly attached to the outside of the cell wall or loosely attached. If it's firmly attached, we call it a capsule. And if it's loosely attached, we call it a slime layer. Now there are three main functions of this glycocalyx. As you can see from the slide, one of them is adherence, so it's nice and sticky to help it adhere, meaning the bacteria can better adhere to your tissues, to your cells, or to surfaces outside of your body. It's also a great protective structure. Okay, so as a capsule or slime layer, it's protecting the bacteria from the outside world, and it protects from desiccation. Now, desiccation, you'll hear again later in the semester, that's basically a fancy way of saying drying out. The third function of a glycocalyx is that, think about it, if you have this structure on the outside of the cell that's basically highly made up of sugar compounds or carbohydrates, during desperate times, if you think of your own body, what you can do with, sugar sources or fat sources, you can start to break them down as a source of nutrients. And that's exactly what bacteria can do with the glycocalyx. So during stressful, desperate times, they can use the glycocalyx for nutrition. Okay, so that's the first structure here on this list. Some cells also have fimbriae or pili, which both help with adherence. Fimbriae are basically look like little hairs, and they will help the bacteria to stick to things. Pili can also be for adherence, but they're also for movement, specifically gene transfer. So during conjugation, which we'll talk about in a different lecture, which is basically cell sex, bacteria can use pili to transfer DNA from one bacteria to another. Now, in addition to the ones that we have for adherence, which are so important because remember, adherence is part of those 5 steps for a pathogen. If you remember, bacteria has to enter, stay in, destroy defenses, do damage, and transmit to a new host, and adherence is basically staying in. In addition to adherence, some structures on the outside of the cell wall are very important for movement. So flagella and axial filament are very valuable to bacteria as they can give the bacteria what we call motility, the ability to move on their own. So flagella, if you've ever seen that in pictures, you'll get to see it on the next slide too. That's basically like having a little propeller. And there's various types of flagella, which you'll go over in lab, that can help the bacteria move on its own. There are also axial filaments, which I want you to remember the term corkscrew. Okay, axial filaments will be on spirochetes that have a corkscrew shape, and the axial filaments not only help them move on their own, but they help them to embed into your tissues as well. So now, in case you missed writing down the notes on glycocalyx, I took a minute to write them all down here so that you can jot them down in your notes. You can pause the video if you need extra time to write them down. Again, just remembering that the glycocalyx can either be a capsule or a slime layer, and that it has 3 main functions, which are allowing it to adhere to things, protecting the cell from desiccation or outside forces, and serving as a source of nutrients in desperate times. Now, to help you visualize some of the structures that we just talked about, you have the glycocalyx over here. So if it's very tight around the cell, it's considered a capsule, whereas loose, it's considered a slime layer. And one of the places that you tend to find bacteria that have a glycocalyx around them is plaque on the teeth and various other biofilms, which we'll talk about that more in other lectures. Biofilms will keep coming back. We also mentioned on this slide fimbriae, which are these yellow structures here that look like a bunch of hairs sticking out from the bacteria's cell wall. You have pili or a pilus over here connecting two bacteria for the movement of DNA from one to the other. You have flagellum, a flagella right here, looks like a little propeller or a whip that the bacteria can spin around to help push the bacteria forward. And you also have the axial filament, which you'll find on spirochetes that helps them gyrate or corkscrew into things. And an example for the axial filament will be the bacteria, the spirochete that causes syphilis and allows syphilis to embed within tissues. Now, whenever we talk about the cell wall or cell structures, if you were to just take a bacteria and plop it on a slide and try and look at it under a microscope, you wouldn't be able to see anything. You need to stain the bacteria to better visualize it and to help distinguish the various structures or characteristics of that bacteria. And you're going to get to do the staining in lab, of course, but this is to give you some background information. So there are two categories when it comes to stains. There are simple stains and there are differential stains. When we talk about simple stains, a simple stain is simple because it is only one dye. So for instance, if you just have methylene blue and all of your bacterial cells on a slide turn blue, that's a simple stain. Now, since you're filling all of the bacteria with the same color, you can't tell too many things from a simple stain. All you can tell is size, shape, and arrangement. If you want more information on that bacteria cell or to be able to distinguish things, then you need a differential stain. And what a differential stain is exactly what it sounds like in its name, different colors. You now have two or more dyes, which means now you can distinguish between different structures or different organisms, which is very valuable, especially in the medical field. Some examples of differential stains are the Gram stain, the Zeolnisen acid fast stain, and a negative stain. The one that you'll be doing in lab soon is the Gram stain. And the Gram stain allows us to differentiate cells based on cell wall composition. So basically, is the cell Gram positive or is the cell Gram negative? Now, if you look at the figures over here, this figure here is an example of a Gram stain. The purple cells are Gram positive, whereas the pinkish looking cells, those are Gram negative. And when you look at this picture, you can ask yourself, what shape are the Gram positive cells? Well, they look round, so they are coccus shaped. Whereas the Gram negative cells, what shape are those? They are bacillus, okay, rod shaped. Okay, so always practice some of the materials that we covered in previous slides. The second example of a differential stain here is the Zeolnisen acid fast stain. And I want you to put a star next to that and write down TB, as in tuberculosis, and leprosy. So the Zeolnisen acid fast stain, in that stain, if a cell becomes red, which I know in this picture it doesn't quite look red, but if it's red, kind of like coughing up blood or blood of limbs coming off, that tells you it's positive as a mycobacterium, which would be either TB or leprosy. Okay? And they're called mycobacterium because they have mycolic acid in their walls. Please do not get them mixed up with mycoplasma, which have no cell wall. Okay? And then we have the negative stains, which are the capsule stains. And in this stain, instead of staining the cells right away, you stain the background of the slide, And then you put a second stain inside the bacteria. So you can see which bacteria have a capsule because they have that halo around them. All of these stains, though, you'll notice you're able to distinguish or differentiate either different cells or different structures. So in case you had any difficulty writing down what I was talking about on the last slide with regard to the acid-fast stain, here are some notes I wrote down for you to jot down. You can pause the video in case you need a little more time to write them down. It's basically just summarizing the idea of an acid-fast stain, demonstrating if a mycobacteria such as TB or leprosy is present. Red cells are a positive, meaning yes, the TB or leprosy is present. Blue cells are a negative. And like I said, make sure you do not mix these up with mycoplasma, even though they sound very similar. Okay, so one of the most important stains we have in microbiology, and like I said, you perform this in the BI 219 lab, is the Gram stain. And the Gram stain allows us to distinguish cells based on their cell wall composition, meaning are they Gram positive with plenty of peptidoglycan and very few lipids, or are they Gram negative with all of those extra layers, thin peptidoglycan, and a whole lot of lipids. So to the Gram stain, when you perform this differential experiment, you have 4 main reagents, and I want you to know the purpose for each one and how any mistakes with them would then change your results. So first, you add a crystal violet primary stain. And when you hear primary stain, that means it's the first dye that you're adding to your cells. What happens here is all cells take in the primary stain, meaning both Gram positive and Gram negative will take in whatever the primary stain is that you use. If you use crystal violet, then all cells at this stage will be purple. So Gram positive and Gram negative after the primary stain of crystal violet are all purple. Keep in mind, some people may not use crystal violet, so whatever your primary stain color is, that's what all the cells become. You then add an iodine mordant, and what a mordant does is it complexes with the primary stain. And what that means is it's now making that stain much bulkier to help trap it within the cell. So it helps keep that color in the cell so that it's not easily leaking out. You then add 95% alcohol wash, which is a lipid solvent. I want you to put stars or circle or highlight that fact that the alcohol is a lipid solvent, because where did you hear lipids? Which cells? The Gram negatives. Only the Gram negatives have a ton of lipids, and if it's a lipid solvent, that means alcohol will dissolve lipids, which means that it's starting to break down the Gram negatives cell wall. Okay, so if it's breaking down the Gram negative cell wall, what happens? The primary stain color leaks out now, but only from the Gram negatives. So at this step, what do your cells look like? Gram positive is still purple, or whatever your primary stain color is, whereas Gram negatives have lost their color. They are colorless. It leaked out. You then add the final reagent, which is the counter stain. In this example, it's safranin, which is a pink color. The counter stain can only enter any cell that's currently colorless, because think about it. If you try and put the counter stain in those Gram positives, they're full of dye already. There's no room for a counter stain. So instead, the counter stain will only enter empty cells, Which cells are currently empty or without a stain right now? The gram-negatives. So at the end of this stain, the gram-positives still have the primary stain, whatever color that was, which in this example is purple, and the gram-negatives now have the pink color, the safranin. Now you have to ask yourself, what are some things that can go wrong? And what would the cells look like if any of these errors happen? This you'll have to practice in lab and in lecture. So on the next slide, I'll give you a little practice. So here are some examples of what I was talking about when I said gram stain problems. Okay, so when you are kind of studying gram stains, to determine if you truly know the meaning of the gram stain steps and the function of each reagent, you should be able to troubleshoot if any of those reagents were forgotten or swapped. Here are just two examples, but there are many other ways that I can ask these. The first one says, if a student accidentally swapped the crystal violet and the safranin, what would happen? So that basically means that they did safranin as the primary, then iodine, alcohol, and crystal violet. So whenever you do any of these problems, go step by step. Okay? There are 4 steps that you should normally be saying, and ask yourself what happens after each of the steps now. So if they first added safranin, which is a pink color, what happens with the primary stain? Well, it goes into all cells, including the gram-positives and the gram-negatives. So all cells would be pink at the beginning. Iodine would then help trap that color in the cells. Alcohol would then do what? Dissolve the lipids of the gram-negatives. So now the gram-negatives will lose the pink color, and what would enter them? The crystal violet. So in this scenario, now the gram-negatives would be purple, and the gram-positives would remain pink. In the second example, we ask, what if they forgot the alcohol step? If they forgot the alcohol step, once again, go step by step to what they're doing and see what would happen. So they remembered the first step, which is normally crystal violet, so all of their cells turn purple. They then add iodine, which is the second step, and that helps trap the purple color in. But now, without the alcohol, what doesn't happen? Well, gram-negatives never lose their color, so everyone still has purple inside of them. So what happens when you try to add safranin or a counter stain at the end? Can't enter any of those cells, right? Because all of those cells are still full. So at the end of this gram stain, everyone's cells, both gram-positive and gram-negative, would be purple. And so this would no longer be a differential stain, but would instead act like a simple stain. If you want any extra practice problems for gram stain issues that can occur, just ask me in Remind or through email, and I'll give you more practice, especially before the next exam, which will cover this material. Now, in addition to the cell wall, there's also the plasma membrane, which is right below the cell wall. Now keep in mind, I mentioned our cells also have the plasma membrane. So even though we hear it constantly said that the plasma membrane has no pathogenic qualities but may be a target for therapeutic strategies, make sure to write next to that it's not a good target, that usually we do not want to target the plasma membrane because we can hurt the patient as well. What is important about the plasma membrane is that it greatly affects the permeability of cells, meaning it guides what can enter a bacteria and what can exit a bacteria. It also houses the electron transport chain, meaning that it's part of how bacteria can make their ATP, or their energy. The last thing to note here is that the plasma membrane is very important for transport, and you have to know the difference between osmosis versus diffusion, which are passive and active diffusion. When we talk about these 3 types of transport, The first one is osmosis, and I like this little figure here for osmosis. Okay, that demonstrates that osmosis is the movement of water. Okay? And when the water is moving, it goes based on low solute to high solute, which is a fancy way of saying from high water to low water. Now, when you see that movement of water, okay, if you have a hypotonic solution, hypotonic means low salt, right? Low salt around that cell. So if water is moving from where there is low salt, which is around the outside, then it's going to be moving from the outside to inside the cell, which is why in the figure here you see the cell swells and then can burst. The way that I remember what happens in hypotonic solutions is I think hippo, hypo. So whenever you hear hypo, think hippo, getting really, really big. The cell gets really, really big, it swells and can burst. The opposite is true in hypertonic solutions. So hypertonic solutions, which is this guy here, if it's hypertonic, that means there's a lot of salt on the outside of the cell. Whereas in comparison, the inside of the cell is lower salt. And as we just said, the water moves from low salt to high salt. So in this case, it's exiting the cell. Okay, so the cell shrivels. To help you remember what happens in hypertonic solutions, always think of salty environments, right? If you eat a ton of salt, what happens? Your mouth is all dried out, right? You get that drying out, kind of like the shriveling cell. Or if you've ever heard of what happens when kids pour salt on slugs, they shrivel up. Okay, so you get the loss of water. The ideal, which for all of our nursing students or anyone going in the medical field, is isotonic. Isotonic means there's a balance inside and outside the cell. So the water is not going to be lost or gained in any high amount, which is what you want because you don't want cells bursting or shriveling up. So you'll notice in hospitals, a lot of the solutions that you use will say isotonic buffers. The other types of movement that you can have from a cell's plasma membrane is either active transport or passive transport. When you think about these terms, always ask yourself, what does active mean? What does passive mean? So if something is active, that means it's high energy, right? So active transport requires a lot of energy, which means that it's movement against how cells normally want to move things. So if cells normally want to go from high to low, right, because they want to equal things out, active transport will move things from low to high gradients. Okay, so low solute to high solute. And this now, instead of the water moving, this is the salt or the solute themselves moving. Passive transport, on the other hand, is passive because it doesn't require energy. It's exactly the movement in the direction that things normally want to go, which is from high to low. Okay, because high to low would then balance things out. Okay? So keep in mind, again, osmosis, what happens to a cell in each of the three conditions. Make sure you know that. And know the difference between active and passive transport. So the idea that active transport requires energy, which is because it's going against the normal concentration gradient, because now it's moving things from low to high, versus passive transport, which does not require any energy because it's happily going in the proper gradient direction of high to low. So the last of the structures outside of the cell wall that we'll talk about before moving on to internal structures are endospores. I like to call endospores little escape pods for certain bacteria. Now keep in mind, only certain bacteria are capable of forming endospores, but the ones that do, these endospores make them very dangerous to us. So what an endospore is, you can see it in this figure here. It's basically a protective structure. So these certain bacteria, what they can do is they can undergo sporulation, meaning if the environment around them is suddenly very dangerous, they can protect themselves from it by leaving their vegetative active state and instead forming an endospore that is a double capsulated structure with just DNA inside. Okay? This protects them from the outside environment, which at the moment is very unfavorable to them, either due to things like very high heat or toxic chemicals or UV, etc. So in this protective endospore, they are now highly resistant. What they then do is they stay in this state of dormancy, meaning inactive, while the threat is there. And then when conditions start to get more favorable, for instance, if the temperature has now dropped to a very comfortable temperature, or if the toxic chemicals have gone away, what they then do is they germinate. And what germination means, just kind of like picturing a bulb coming out from the ground as a beautiful flower, what these guys do for germination is their outer capsules of the endospore will crack and allow water in, and the cell will then return to its nice big vegetative active state because conditions are better. Okay? Now, keep in mind, I mentioned before that endospores are very dangerous to us because of the fact that they're so highly resistant to all of the things that we usually use to try and kill bacteria. So things like high heat, desiccation, which we said before is drying out, toxic chemicals, antibiotics, UV, and even boiling. If a bacteria can produce endospores, they can survive all of this. The big clinical example to remember is botulism, which we'll talk more about in later chapters. But botulism, for instance, most people associate with canned products, and the reason why botulism survives the canning process is because it can go into endospores and stay dormant until the canning process is over, and that can is now sitting on a nice, comfortable, perfect temperature, perfect environment shelf in your house or in the store. Okay? So again, we'll talk more about this in later chapters, but for now, I want you to remember whenever you see endospores, think extremely resistant. Remember sporulation, meaning they form endospores when the environment is stressful. They are dormant, and then they germinate when the environment is better. Okay? And keep remembering that example, botulism. So that's it for the outside anatomy terms. Now we're going to go through some internal anatomy structures of bacteria. The first internal structure to remember is cytoplasm, and we also have cytoplasm. There's two terms that you'll hear associated with this. Sometimes you hear the word cytoplasm, whereas sometimes you hear the word cytosol. The difference is that cytosol is simply the fluid within the cell, whereas cytoplasm is the fluid plus all of those dissolved suspended particles such as proteins, carbohydrates, lipids, and ions. Okay, so when I wrote here, this first bullet point here, this is the definition of cytoplasm, whereas cytosol would just be the fluid. The other term here to know is cytoskeleton, and the cytoskeleton, just like our skeleton, is for keeping shape and structure. So the cytoskeleton is made up of tubulin-like proteins that help keep the bacteria looking like a bacteria. Another internal structure of bacteria is the nucleoid, because remember, what did we say about bacteria? They're prokaryotes. No means that they do not have a nucleus. Instead, they have a nucleoid, which will resemble the nucleus, meaning it has DNA in it. but it has no nuclear membrane. Within the nucleoid, you'll find the bacterial chromosome, which is a big circular ring of DNA that has all of the cell's genes that are critical to its life functions. Bacteria also have what are called plasmids. So keep in mind, plasmids are extra chromosomal, meaning that they're beyond the regular chromosome, which means not all bacteria will have them, and the bacteria that do have them, they have various different versions of plasmids. What's important about plasmids is even though they're not essential for the life functions of that bacteria, because we said they're extra, just like some people I know, what they can do is they can confer an advantage to the bacteria, and the big advantage that they can give that I want you to remember is antibiotic resistance. So when we talk about antibiotic resistance in lab and in a later lecture, remember that bacteria get antibiotic resistance from plasmids, from genes on plasmids. Okay? So when I wrote what may be an advantage to a bacteria, the answer is antibiotic resistance. The next structure to remember are ribosomes, and you're all experts at the ribosome. Okay, as you know, the ribosome is the structure for protein synthesis, and you also know that it's a nucleoprotein. And again, make sure you remember that term, nucleoprotein. So that means that the ribosome is made up of protein as well as a strand or some strands of RNA. We said that that's very significant because by having the single strand of RNA, that allows the ribosome to grab a hold of other strands of RNA, specifically the mRNA. Okay, so the fact that it's a nucleoprotein is significant because it allows the nucleoprotein, the ribosome, to grab a hold and bind mRNA. You don't have to memorize that it's 70S. That's just to show you that the way they categorize the size or the structure of ribosomes is that terminology there. And I know it looks weird because 50 and 30 does not equal 70, but that simply has to do with a centrifugation type of terminology. But again, you don't have to memorize that. I just put it there in case you ever see that in writing and are curious what it's referring to. The last of the structures to know are inclusions, and I always tease the students saying, oh yeah, you have to memorize every single one of the ones on this slide, but that is a complete lie. All I want you to know is that the term inclusions means structures that hold things. All of the bullet points on this slide are just examples of inclusions, and as you can see, each one can hold different things. So some of them are holding phosphates, some are holding energy, some are holding various enzymes or proteins. But ultimately, the thing to remember is inclusions are simply little sacs that hold things. Okay, so that's it for bacterial anatomy. The last thing I want to talk about in this lecture is some information about mechanisms of pathogenicity. And this is where we start to hit some of the more interesting facts of microbiology because they're much more relatable and things that you'll encounter in the hospitals, in the medical field, or even within your own life when you or your loved ones are ill. The first thing we have with regard to mechanisms of pathogenicity is a slide that you've seen before. Now remember I said you have to know the 5 requirements for a microbe to be pathogenic, to have a successful infection. So for a microbe to be pathogenic, it must be able to enter the body or cell, stay in, defeat your immune system, your defenses, cause some type of damage, and transmit to a new host. So some students remember it as ESDDT. Enter, stay in, defeat, do damage, and transmit. Now what's new on this slide now is the term virulence factors. And I want you to circle star, highlight that term, because virulence factors is something that will come up multiple times throughout this course, and you have to know the definition. What virulence factors means is any molecules that are produced or secreted by the bacteria to do all of these above things. Okay, so virulence factors are any molecules or structures that help bacteria do any of these 5 requirements for pathogenicity. Okay, one more time. Virulence factors are any molecules or structures that let bacteria or help bacteria do any of these 5 things to be pathogenic. So we're going to go one by one through these various requirements for successful infection. The first requirement for a microbe to be pathogenic is to be able to enter the body or the cell. There are various ways that they can enter the cell or the body. Okay? We call that portal of entry, meaning how it's entering, where the microbe's actually getting in. And a lot of times this portal of entry can also serve as an exit as well. So there are 2 main categories of portals of entries for bacteria. The first ones are mucous membranes, okay? So any of the tracts that you see listed here, such as respiratory, gastro, genitourinary, conjunctiva, all of these have holes adjacent to the external environment and allow for microbes to get in. So when you look at these tracts, there are a few things you have to ask yourself. First of all, which of these tracts is going to be the most common and most problematic portal of entry? Well, that would be the respiratory tract, because think of it, you always have to breathe all day, every day. The gastrointestinal tract will be the second most problematic portal of entry because you always have to eat or drink. But at least with that one, it's not every second of the day, the way breathing is. Genitourinary is becoming more and more problematic as a portal of entry, and we'll talk more about that in the STD lesson. But for now, if you think about it, it's not, even though it's very, very high rates and problems today, when you look at it compared to the other tracts, people don't have to have sex 24/7 the way they have to breathe. So it's not as common as respiratory, taking in microbes. The last one is conjunctiva, and conjunctiva, right next to that, write the word eyes. Okay, conjunctiva means your eyes. And we'll go through in a later lesson how your eyes actually do a lot of things to prevent microbes getting in this portal of entry. Okay. The other big portal of entry is the skin. And any bacteria that enters through the skin, through breaks in the skin, is considered as entering through the parenteral route. Now, when we think of the skin, remember the skin is the largest organ of the body. And we're going to talk about the skin and the immune system as being very good at protecting us. But even though it's very good at protecting us, a lot of microbes end up penetrating the skin barrier and entering through the parenteral route entry. And again, parenteral route simply means that a microbe enters your body through a break in the skin. So examples of that would be any kind of cut, puncture, injections, having surgery, or even things like insect or dog bites, all of which we'll go through in various lectures throughout this course. The big thing is make sure you know terminology, what it means to be a portal of entry, what the examples of portals of entry are, and what the term parenteral route means. So once a microbe has entered our body, a lot of them now want to try and enter our cells. But if you think about it, each of your cells has that plasma membrane, which is like a barrier, kind of helping to block the pathogen. But many of the pathogens have ways to then break through this barrier and make it So what a lot of them do, especially viruses, is they use specific receptors that are on the outside of your cells. And what they can do is they can then basically bind to that cell, merge with that outer membrane, and then insert themselves inside of the cell. Kind of like you see in this figure here, where the virus has bound to the receptor and then merged with the membrane and popped itself right inside of the human cell. What they then do is when they break free of that cell later on, they burst through or lyse that cell and they steal part of that cell's membrane. And they basically wrap themselves with a piece of the host cell, meaning you, your cell's membrane around it to then travel through your body. And you kind of ask yourself, well, why would a virus want to do that? Why would a virus want to wrap itself in this extra envelope that's basically part of our cells? Well, think about it. What's on that outer envelope? What did we say was on there? Receptors. So now that virus can travel around in your body, basically wearing a coat that looks like one of your cells, and that has the receptors to match your cells, which makes it a lot easier for that virus to escape your immune system and also to infect other cells within you. So if you ever see a virus that has an envelope, because we're going to mention them in later lectures, that means that it stole that membrane from one of the host cells. Basically, we say that viruses are jerks. Now, once the pathogen has entered your body or your cells, what's the next step that it has to do to be a true pathogen? Well, it has to stay in. Because think about it, if you instantly coughed or sneezed it out, it's not going to do any damage. It's not going to be a problem or a pathogen. So we kind of touched upon some of the structures that certain bacteria or microbes have that help them to stay in. Okay, so we mentioned fimbriae, which are these hair-like extensions here, which help them to stick to things such as your cells or tissues. A lot of bacteria also have what's called adhesins. And adhesins, which you see in the figure here, they're simply surface proteins, basically receptors, that help them grab a hold of receptors or proteins that are on your cells or tissues. A big example of this adherence using adhesins are the bacteria that make up plaque on your teeth. Okay, they also tend to have glycocalyx, which we mentioned was that sticky capsule that also helps them to adhere to things. So just to kind of recap one of the big terms that I said you would have to remember, if we say that fimbriae and adhesins or the glycocalyx, any of these structures are meant to help the bacteria stay in, which is one of those 5 characteristics of a pathogen, then what do we call them? They're virulence factors. Okay, so remember the term virulence factors. Fimbriae, adhesins, glycocalyx, any of those are examples of virulence factors. They help the microbe to be a pathogen. Now, when we talk about staying in, another example of how bacteria can stay in is the example of spirochetes, such as treponema, which is one of my favorite of the giant plushies that you'll see that are present in the lab. Treponema is spirochete, which is why it has that spiral shape. It's what causes syphilis, which we'll talk about in more detail in the STD lesson. But what's important about this guy here, syphilis or treponema, is that it uses axial filaments, which we mentioned in an earlier slide. The axial filaments let the treponema, or causing syphilis, embed itself or corkscrew into the human's tissues. and organs to really attach, to stay in. Okay, so whenever you hear syphilis, think of axial filaments. And that's how it is motile, and that's how it establishes or attaches to different tissues. Now, when we talk about microbes attaching, adhering, getting established within the human body, the number of microbes that actually establish themselves determines how successful an infection is present. Now, the number required for this kind of success varies, and there's two terms that I want you to be familiar with, especially if you go into the medical field, because you'll notice in case studies or various documents, you'll frequently see the term LD50 or ID50. What these are lethal dose 50% and infectious dose 50%, okay? So we'll first give the straight, textbook kind of definitions, but then I'll kind of give you a little insight into how to really work with these values. So the official definition for lethal dose 50% or LD50 is the number of organisms needed to kill 50% of the host. Infectious dose 50%, on the other hand, is the number of organisms, meaning pathogens, needed for 50% of the population to show symptoms. Okay? So now the way to really understand these terms is to think about what it means if you have a low version of the term or a high version of the term. So for instance, if for a particular bacteria, let's say, we just mentioned syphilis, let's say we were told that syphilis has a low LD50 and a low ID50. What would that tell you about the syphilis pathogen? Well, if it's low LD50 and low ID50, that means it only takes a couple of this bacteria, picture, one or two of this bacteria and boom, half the people die. Or one or two of this bacteria and boom, right away 50% is showing major symptoms, brain dysfunction and severe illness. Well, if it only takes a couple of them, a low number of them, to make people so sick, well, that tells you that each individual one must be really, really strong, right? Whereas if there's a high LD50 or a high ID50, what would that tell you? Well, you need millions of this little bacteria before people even sniff them. Or you need millions of these just to kill one person. Okay? That would tell you that each individual one isn't very strong, right? So for the first question that I have highlighted in red, this one here, low LD50 and or a low ID50 tells you the pathogen is very virulent. Okay, so for this one, write very virulent. Whereas a high value would tell you not very virulent. Okay, low virulence, not a strong pathogen. Okay. So make sure you're very comfortable with those two terms, and if you need a little clarification, just send me a remind message or an email. Now, once the pathogen has entered and stayed in, the next thing it must do is defeat the defenses. And remember, we said that basically means defeating whatever your immune system is throwing at it to try and kill it. Now, when we talk about defeating the defenses, you have to know the difference between passive and active strategies. So if something is passive, remember, active means energy, whereas passive means no energy. Very passive, you don't need to do anything extra. It's just there, right? So for bacteria, if they're trying to defeat the host defenses passively, it tells you that it's something about them that they don't have to do anything extra for. They already have it. So passive would be built-in structures on the bacteria to protect them, such as capsules, which can physically block what you're throwing at it, or help prevent any of your immune system cells from eating them up, which is phagocytosis. or their cell wall, which again physically blocks your immune system's targeting them, but also has proteins that helps them to do what they need to do in your body, and that can be toxic or dangerous to your body to help protect them further. Okay? So again, passive strategies are parts of the bacteria that they already have to protect them. They don't have to do anything extra. Whereas active strategy, if you are active, you got a lot of energy, right? You got a lot of movement. So for active strategies, now the bacteria is going to be doing something active. It's going to be producing something, secreting something. You got movement here, right? So active strategy would be any of the enzymes that they produce and release to kill off your immune cells. The three big examples of active strategy are leukocidins, hemolysins, and coagulase. So leukocidins, that's an enzyme that will kill leukocytes, which means white blood cells. Hemolysins instead, hemo, are now going to kill red blood cells, causing those red blood cells to lyse. And then coagulase, I'm sure you've all heard the term coagulating. Coagulase will cause blood clots in the host, which slows down those immune cells from getting around. Okay, so make sure you're very comfortable with the term passive versus active and what the examples are of both of these. This slide here is just to show you a picture of what we were just talking about in terms of passive. So passive strategies having the capsule or the cell wall around the bacteria, okay? And active being anything getting secreted in any of these, okay? But again, you never have to memorize any of these figures. They're just to help you understand or visualize what we were just talking about. Now, the last thing that I want to mention over here is the idea of quorum sensing and biofilms. Okay, both of these play a role in pathogen virulence, being that quorum sensing, if you picture this, what this is proteins basically will not be activated and released unless the bacteria assesses the population around it and determines, hey, I have a lot of my buddies around, so now let me activate these genes and produce these proteins and these virulence factors. Now, why would a bacteria want to wait until it senses the environment around it and knows that there are a lot of other bacteria around it before it starts activating genes and producing proteins? Well, think about it. When we went over genetics and gene expression and translation after that, it's a lot of work. There's a lot of enzymes involved, a lot of steps involved. It takes a lot for a bacteria to do these things. So why would you, if you're a bacteria, waste your resources, your energy, your enzymes, and honestly your time producing these proteins and these virulence factors if you're just one little bacteria and you're not going to be able to do much with that? Why not wait until you have a whole big population around you, a whole bunch of bacteria friends, where now if you all use up that energy and resources to produce the virulence factors, now it'll do something. Now the host will actually feel it and you'll accomplish something. Okay, so quorum sensing, the reason they do that is to conserve resources until they know that there are other bacteria around to help complete the task of hurting the host. In addition to quorum sensing, another thing bacteria can do in order to help with being pathogenic and virulence is to produce biofilms. Okay, so certain bacteria will produce biofilms. What this means is they will form a collection or an aggregation of different species of bacteria that stick together and will grow as one big mass. Okay, they're all working together, sticking together. Now, if you think about this, this is kind of valuable to these bacteria because now you have a whole bunch of species of bacteria with you. Okay, so you can obtain different nutrients, you could get different things done, and most importantly, when the host tries to damage you or destroy you, well, if you think about it, a lot of the things that we do to destroy bacteria, it'll get rid of one type, two types, but it's not going to get rid of every single possible type at once. So by having a variety of different species working together, they increase their odds of survival because you can't kill them all at once. So biofilms become very tricky for us to destroy or clean up. And some of the big examples of that, the one that you will constantly hear this semester, and I want you to remember, is plaque. Plaque on your teeth is a biofilm. And that's why it's so difficult to deal with, and that's why you'll have plaque for your whole life. You can never 100% get rid of it. You can minimize it, but you can't get rid of it. Now, the other area that we commonly see biofilms is on medical devices, especially things like catheters or equipment, medical equipment. Okay, and that's why you always have to be very careful in the medical field and try and minimize the chances of any biofilm collection. You also see biofilms in things like pipes and whatnot and sewage, but the big examples that I want you to remember for our purposes are plaque on the teeth and biofilms on medical equipment, such as catheters or IVs. Now, the last of the things we're going to mention today is how pathogens will try to damage the host, okay, damaging the host. Now, with damaging the host, there are two big causes. The damage can either be because of the pathogen itself being present and active, or the damage to your body can actually be because of your own body, because your immune system is trying to fight those bacterial cells or those viruses and accidentally, friendly fire, injure your own cells or damage your own cells and tissues. Okay, so if you think about things like excess inflammation, your immune system's trying to help you, but it can also be quite problematic if you have too much inflammation. So when we talk about damaging the host, there are two big categories to remember. There's direct damage or indirect damage when we're talking about damage being caused by the pathogen itself. Now, if it's direct damage, then you're basically going to think of it as the direct damage from the physical pathogen itself. So kind of picture if that pathogen is kind of crashing into cells and tissues. So the pathogen itself is destroying host cells or tissues. Okay? Indirect damage is now when you have a systemic, so body-wide trouble because of toxins produced by the pathogens. And in the next couple of slides, we're going to talk about the two big categories of toxins, and you have to remember these toxins, okay? So you are going to circle, star, highlight toxins, okay? So the two categories of toxins, again, putting stars all over this slide and the next slide, the two big categories of toxins are exotoxins and endotoxins. So when you hear the word exo like exoskeleton, that tells you it's on the outside. It's released. So exotoxins are secreted to the outside of the cell, of the bacterial cell. The fact that it's secreted and it's from living bacteria, this is very problematic to us. And it makes them the most lethal of toxins. Okay, so circle the fact that these are the most lethal and put a little star next to living bacteria secreting. Okay, the reason these are most lethal is because think about it. These toxins can act remotely without the pathogen, and if you kill the bacteria, that's not going to be enough to heal the patient. You still have these toxins floating around through their body. Now, what is most lethal about them, though, is the fact that these are toxins being secreted by living bacteria. They are still alive, which means they can continuously pump more and more and more toxin throughout your body. That's more lethal and damaging to you because it's a factory basically, consistently pumping this toxin throughout your body. You'll see in a minute why this or how this varies from endotoxins, which are not as toxic. Now, the big examples of exotoxins that you tend to encounter are cytotoxins, neurotoxins, and enterotoxins. Whenever you hear the term cyto in biology, cyto means cells. So cytotoxins will kill any cell that it comes in contact with. A big example of this is anthrax. So if you heard all those times there were anthrax scares, that's because the anthrax toxin will be cytotoxic and it will cause major hemorrhages and pulmonary edema as it's destroying cell after cell. Neurotoxins, another example of exotoxins, are also very lethal because these guys interfere with your neurological signals. So a big example of this that I want you to remember is botulinum toxin, so the toxin from botulism, and remember tetanus. So botulism and tetanus will produce neurotoxins. Since they're targeting the neurological signals in your body, that means that they'll block things like acetylcholine and cause paralysis. Now you may ask yourself, well, how is paralysis lethal? Well, it's not just muscles, regular old moving your arms and legs that becomes paralyzed. What other muscles can become paralyzed? Cardiac and respiratory. So you lose the ability to pump blood or to breathe, so the patient dies. So neurotoxins are usually rather fast-acting and dangerous. The last one on the list here for exotoxins are enterotoxins. And whenever you see the word entero or enteric, remember GI tract. So enterotoxins will affect the lining of the digestive system and cause things like diarrhea and vomiting, which can be dangerous in terms of dehydration. Now, the alternative is some bacteria instead will produce endotoxins. And when you hear endo means within. So endotoxins are within the bacteria's own cell wall. They're in the outer layers of gram-negative cells usually. So for instance, the lipids that we mentioned in earlier slides. The difference with endotoxins, since they're built into the bacteria's cell wall, endotoxins are only released when the cell dies. This means that they're not as toxic as exotoxins, because think of it, what's more dangerous? If you have a serial killer or if you have someone who commits suicide when they commit the murder? Well, the suicide killer, they only can kill whoever they killed at that moment. They can't kill anymore once they're dead. Whereas a serial killer, which is the exotoxins, they're still alive. They can keep killing, keep releasing toxins. So endotoxins are not as toxic because they're basically suicide death. Okay? So endotoxins are less toxic and released only when they die. And the other thing that they're most known for is causing fever. Okay, fever. Now, one last bullet point that I mentioned here is that endotoxins are more heat stable. And that makes sense since we just said that they target fever, so it makes sense that they're able to withstand the heat. It's basically because these are lipopolysaccharides usually, whereas exotoxins are soluble proteins that can end up denaturing. Okay? So the last thing I want to show you is when we say trigger fever, this is what we actually mean. So with endotoxins, what happens is you have a bacteria here and it gets engulfed by one of your immune cells, a macrophage that's known for phagocytosis, which is trying to engulf and break down the bacteria. Your immune cell thinks that it's protecting you and doing good by breaking down this bacteria and killing it for you. What it doesn't realize is that this bacteria, usually a gram-negative cell, once broken down, as you can see here, the little pieces of the outer membranes break apart, break down, and those happen to be endotoxins. These endotoxins then induce cytokines, which are these little chemical circles here, and the cytokines then travel and activate the hypothalamus. The hypothalamus will produce prostaglandins, and that's basically the thermostat in your body and causes your temperature to increase, okay, to give you a fever. A fever is actually part of your immune system, and fever is meant to do good for you. We're going to talk about this in a later lecture, but for now, just ask yourself, well, why would a really high temperature be good? Well, bacteria, they're not going to like that high temperature. So it's trying to help reduce how favorable your body is for the bacteria. Now, again, this is just to show you kind of how endotoxins work, but you don't have to memorize this figure. Just be aware that endotoxins are the type of toxin that will induce fever and that are not as lethal usually as exotoxins. I really want you to remember exotoxins as the big bad guys. Bad like an X. Thank you, and make sure to send me any questions as you study, either in the Remind app or an email. And if there's any figures or particular slides that you need extra assistance with, feel free to send me pictures of them or to list out various questions. You can number your questions, and I'll reply with numbered responses so that you know exactly which question I'm targeting. And as I always say, I have no life, so feel free to ask me as many questions as you need. I'm always here to help.